Let's talk about the synthesis of the thyroid hormones. Now, the synthesis of thyroid hormones is a simple one. The thyroid hormone, the both of its lobes, the right and left, and the isthmus, all of which are composed of follicles. Now, these follicles contains follicular cells. As you can easily see here, that both of these lobes have these follicles in them, and these follicles have follicular cells which are actually simple cuboidal epithelial cells, but they are special cells because they take up the iodine from the bloodstream and then convert it with the help of thyroglobin into the two iodinated hormones, and these hormones being the T4 and the T3 hormones. Now, it is worth noting here that it is the T3 hormone that is the active component of the iodinated hormones group, but it is released in the minor quantity. It is T4, which is usually inactive form. It circulates more widely in the bloodstream, but when it enters the cell, it is transformed into the T3. Now, how it happens, why it happens, we will discuss it while discussing the regulation of the thyroid hormones. Now, the thyroid hormone synthesis is a complex one because there are two main bodies which dictate on the thyroid gland that how much of the thyroxine and T3 they are allowed to release into the bloodstream. Now, these two bodies are the hypothalamus, which is this part of the brain, and the anterior part of the pituitary gland. And because of their influence on the thyroid gland, it is what is known as the thyroid-pituitary-thyroid axis, because all of the process works in a flow-like manner. So it goes from the hypothalamus to the anterior pituitary, then to the thyroid. So first of all, we're gonna start with the hypothalamus. Hypothalamus is a small part of the brain which lies at the floor, and it is attached with the pituitary gland of the brain, which lies in the sphenoid bone. And when we look at both the hypothalamus and the pituitary gland, we get a more clearer picture at what is happening. As you can see that this is the hypothalamus. Now this hypothalamus is the one that lies at the floor of the third ventricle. Now this is the third ventricle, and this whole area is the floor of it. And hypothalamus lies at the floor of the third ventricle. And hypothalamus in actuality is composed of multiple nuclei. There are many nuclei that perform a variety of functions. But we are only concerned with one nucleus here, the paraventricular nucleus. The paraventricular nucleus has generally two types of neurons. One is the parvocellular neurons, the other are the magnocellular neurons. As you might recall from the lecture on the posterior pituitary, the magnocellular neurons are the ones which synthesize and release the oxytocin hormones, while the parvocellular neurons in the paraventricular nucleus are the ones which synthesize a certain chemical known as the TRH. Now, this TRH, or the thyrotropin-releasing hormone, it is synthesized in the cell bodies of the parvocellular neurons, and after it is synthesized, it moves in the exoplasm along the axons towards the median eminence. Now, the median eminence is the most inferior extension of the hypothalamus. And at this point, the axons form their end. And at this site, they start to release all of their formed hormones into the bloodstream. Now, all the hormones which are being synthesized as oxytocin, they are actually moved towards posterior pituitary. But all the axons which carry the TRH or the thyrotropin-releasing hormone are released at the median eminence. Now they are released into the median eminence because there is a certain web of vessels here known as the hypophyseal portal system. Now, this hypophyseal portal system that you might recall from the lecture on the pituitary gland is composed of superior hypophyseal artery, that is this one, and the inferior hypophyseal vein. Now, because of these two webs of blood vessels, which are sinusoidal in nature or they are fenestrated, meaning that they have huge holes in them. So, anything that is released at this end, at the median eminence, gets immediately taken up by the web of the blood vessels, which is found here. And because of this, the TRH, which is released at the median eminence, moves through this web of portal system downwards towards the infundibulum or the stalk of the pituitary gland into the anterior pituitary. Now, anterior pituitary again has the sinusoidal web of the blood vessels, and it contains a variety of different cells. And these cells are differentiated enough that they produce different types of hormones. But here we are only gonna talk about the thyrotropes. Thyrotropes are the cells which synthesize the TSH or the thyroid stimulating hormone. So, when TRH is synthesized in the cell bodies, moves through the exoplasm, released in the portal system, moves towards the pituitary stalk into the anterior pituitary, gets in touch with the receptors on the thyrotropes, it signals the thyrotropes to release the TSH that is formed inside their cytoplasm. And once they release their TSH or the thyroid stimulating hormone into the bloodstream, it moves through the sinusoids and out into the main bloodstream. So, in order to simplify this process, we can easily say that the hypothalamus releases TRH, and this TRH is released in a huge amount. And this TRH then gets attached to the anterior pituitary. Now it gets attached to the thyrotrope cells of the interior pituitary. And when it gets attached to the thyrotropes, it releases the TSH. And this TSH or the thyroid stimulating hormone moves through the systemic circulation, and when it gets in touch through the vascular nature or the vascular parenchyma of the thyroid gland, it attaches itself to the cells which are found there. And these cells are known as the follicular cells because they are found inside the follicles, which is what the thyroid gland is made up of. So, inside the thyroid gland, when TSH approaches the follicles, these follicles are like a bag or they are found in a more of a circular nature. This is the follicle of a thyroid gland, and you can see that it is lined with simple cuboidal cells, and these simple cuboidal cells are what is known as the follicular cells. Now there are two types of cells found in the thyroid gland. One is the follicular cells, which are found inside the follicle, and other is the parafollicular cell, which is found outside the follicle. Now, the function of the parafollicular cell is to synthesize the calcitonin. As you might recall, it is the other hormone that is released by the thyroid gland, and it is involved with the calcium balance. So what happens is that inside these follicles, and there is two type of membranes. One membrane is outward facing and one membrane is inward facing. The outward facing membrane is known as the basal membrane, or it is sometimes also labeled as the basolateral membrane. The function of basolateral membrane is to take up the iodine which is circulating in the bloodstream and take it inside the colloid of the follicles, because the follicles are lined with the follicular cells, but they are filled with a colloid solution along with the other organelles of the cell. So what happens is that iodine moves from the blood in through the follicular cells, then the colloid, and then through the other membrane and into the follicle. Now this other membrane is known as the apical membrane, and this apical membrane is the membrane which is found inward of the follicle, and this is the one which comes in contact with the thyroglobin. Now the iodine which moves from the blood all the way through the follicle and into the thyroglobin moves through two transporters. The first one which is mentioned here is the sodium iodine co-transporter. Now, as the name suggests, that both iodine and sodium move in the same direction, and it is an active transport, that it requires energy, that it requires the usage of ATP. And this transporter is sometimes also labeled as NIS, or sodium iodide sim transport, that there is no cross transportation here. For every 2 sodium, 1 iodine or iodide ion is transported from the blood inside the follicle. Once it's inside the follicle, it comes in contact with the iodine chloride antiporter, or in other words, it is the sodium independent exchange. Now, what is meant by the sodium independent exchange? All over the body, whenever sodium is exchanged with the chloride, usually it happens in a crossword manner. Sodium moves inward, chloride moves outward, or the chloride moves inward and the sodium moves outward. This inward movement of chloride is independent of the sodium. Sodium has nothing to do with the inward motion of the chloride. But whenever one chloride ion moves inward, one iodine ion moves into the follicle of the thyroid gland. So, simply put, the iodine from the blood moves through the NIS, through the active transport, into the colloid of the follicles, then through the antiporter and uh, into the follicle of the thyroid gland. Now, once iodine has been transported inside the follicle, two things or two proteins starts to appear. Now, these proteins are the ones which are being synthesized by rough endoplasmic reticulum. Now, these two proteins are, one is the thyroglobin, the other one is the thyroid peroxidase. But we will only see thyroid peroxidase when it gets activated. So what happens is that this rough endoplasmic reticulum start to produce thyroglobin in a huge amount. Thyroglobin is actually a glycoprotein. It is a homodimer, or in other words, it is a dimer of two equal chains. So what happens is that when this thyroglobin is released into the colloid of the follicle, and then it moves, when it gets filled up, it moves outward into the follicles and starts to fill the follicle as a whole. So once thyroglobin is synthesized, it moves. Now it moves through the network of both the endoplasmic reticulum and the Golgi apparatus. And when it moves in large amount. So when the follicle is filled with thyroglobin, the thyroglobin starts to exocytose into the follicle. And once it is out in the follicle, it starts to react with the iodine. So what happens is that because of the thyroid peroxidase, which is produced by the rough endoplasmic reticulum and is transported with the thyroglobin, but is activated only in the lumen of the follicle, starts to iodinate the thyroglobin. Now, thyroglobin, like I said, is a homodimer, and it contains about 100 to 120 tyrosine residues. And it is these tyrosine residues which starts to iodinate. That iodine starts to attach to these residues, the tyrosine residues, because tyrosine, as you know, is an aromatic amino acid. Being an aromatic amino acid, it contains the benzene ring, or more particularly, it contains the phenol group. So iodine starts to attach to the benzene group of the tyrosine, which is found on the thyroglobin. But one tyrosine can only hold two iodine molecules. So, because of this, with the help of thyroid peroxidase, the iodine starts to react with the thyroglobin, and that reaction results in a variety of products. Now, this process where the iodine starts to react with the help of thyroid peroxidase with the thyroglobin is what is known as the organification or the iodination. Now, because of this, what happens is that the tyrosine residues which are found on the thyroglobin starts to get iodinated. That iodine atom starts to attach with them. So, as they start to attach with them, the tyrosine converts into monoiodothyronine, or in other words, it is a simple tyrosine residue with one iodine attached to it. Then another iodine attaches with it and it converts into diiodothyronine. At this point, we have two iodinated molecules. Then what happens is that these tyrosine starts to conjugate or couple with other iodinated residues. Now, these iodinated residues can both be single iodinated or double iodinated. Now, if the residue with which the tyrosine is being attached, if it is double iodinated, it results in T4 or tetraiodothyronine, otherwise known as the thyroxine. But if the tyrosine attaches with a residue which is only singly iodinated, it converts into T3 or triiodothyronine. So, because of the process known as the organification or iodination, the iodine, with the help of the thyroid peroxidase, starts to react together and they result in four types of different iodinated products. And all of these products are different variants of tyrosine attached with the iodine. But this tyrosine is still attached with the thyroglobin. And thyroglobin, being a huge protein, still has a hold or a bond with the said residue. So at this point, we have three steps of this whole process. The first step was the active transport. The active transport of iodine from the blood to the inside of the follicle. The second was the formation of the thyroglobin. Now, thyroglobin is formed by the rough endoplasmic reticulum and it forms the colloid of the follicular cell of the follicles. And when this colloid fills up all of the follicle, it starts to exocytose outside of the follicles or the follicular cells. And because of this exocytosis, we have the fourth process, which I just explained, known as the iodination. And because of the iodination, we have four different iodinated products attached to the thyroglobin. And these four iodinated products need to remove themselves from the thyroglobin. And how they do so is that the thyroglobin starts to couple with other type of residues. That different thyroglobin starts to couple with other thyroglobin. That results in a variety of different iodine combinations, from monoiodine to diiodine, triiodine to tetraiodine. This coupling of tyrosine residue, which is twice iodinated, with a residue with either single iodinated or double iodinated, is what is known as the coupling. And this is what I was talking about when I was talking about the structure of the T3 and the T4. That is, it is important to know that it is only the tyrosine residues which get iodinated. It is not the thyroglobin that gets iodinated. So, because of this peculiarity, it is very easy to remove these iodinated products from the thyroglobin. So, thyroglobin, which has been iodinated or whose tyrosine residues have been iodinated, move inward to the follicular cells. Now, once they move in the follicular cells or the simple cuboidal cells, they move via what is known as the endocytosis. And endocytosis is the process via which this thyroglobin, which has four different iodinated groups still attached to it, moves inward to the colloid of the follicle. And this process is known as the endocytosis. And this endocytosis is the last of the six different steps of the synthesis of the tyrosine iodinated compounds or the thyroid hormones. Now, once because of the endocytosis, this thyroglobin moves into the colloid of the follicle. Different lysozymes starts to cleave it. They start to remove the tyrosine residues from it, and this is what is known as the proteolysis. Now, it is known as proteolysis because it removes the amino acid residues. Because tyrosine is an amino acid, the only difference is that it has been iodinated. So, because of the proteolysis, The tyrosine gets rid of both the monoiodothyronine, diiodothyronine, T3 and T4. So when they are removed from the thyroglobin is regenerated and the iodinated tyrosine residues are free from the thyroglobin, but they are still in the colloid of the follicle. As you can see here, that they are still in the colloid. So only the T3 and T4 move outward into the bloodstream. The monoiodothyronine and the diiodothyronine do not do so. Why? Because they are not the active form of the thyroid hormones. It is only the T4 and T3 that is the active form of the thyroid hormones. Even though it is the T3 that shows the major work of the thyroid hormones, it is released in lesser amounts because T4 is more stable, but because of its stability, it is inactive. So T4 is released as 80% of the total thyroid hormones, while T3 is released as only 20% of the total thyroid hormones. Now, please do note that even though T3 is the active form of the thyroid hormones, T4 is not totally inactive. It still shows some activity in some cells, but it is not as active or does not have the potency of the T3, while the monoiodo and diiodothyronine have no activity whatsoever, and because of this reason they remain in the colloid in the follicle. They do not move into the bloodstream. It is only the T3 and T4 that are moved into the bloodstream. Now when they are moved, the residues which are then found in the colloid are deiodinized. Now, deiodinized means that their iodine residue or their iodine component is removed from them. So once iodine is removed from them, as you can see here, the tyrosine is again regenerated. And because tyrosine is regenerated, iodine is also. So you can easily see here that there are 6 steps to the synthesis of the thyroid hormone. The first one, the active transport of iodine into the follicle. The second, the formation of the thyroglobin. The third one is the exocytosis of both the thyroglobin and the thyroid peroxidase. The fourth one is the iodination. Because of the peroxidase, both the iodine and thyroglobin start to react with each other. And because of that reaction, they start to couple with each other. And because of the coupling, we get a variety of different iodine products, a total of 4: mono, di, tri, and tetraiodothyronine. And when the T3 and T4 are formed, they are then endocytosed into the follicular cell, into the follicle again, into the colloid, where both the mono and diiodothyronine remains, while T3 and T4 moves into the bloodstream. So you can easily remember all of these 6 steps of the synthesis of thyroid hormones by remembering the mnemonic 8 I's. A for active transport, T for thyroglobin, E for exocytosis, I for iodination, C for the coupling, and E for the endocytosis. And after all of these steps, we have the formation or the synthesis of the thyroid hormones, that is the T3 and T4.